you very much. Hi everyone, my name is Katrin. I'm from the Dons Institute in the Netherlands, and for this educational course session, I'd like to tell you about how we can use comparative analysis of diffusion MRI data in order to study brain connectivity. And I don't have any disclosures to report. So a lot of neuroscientific research is focused on the human brain, but we know that in fact the human brain didn't appear out of nowhere, but is in fact the product of thousands of years of evolution. And actually we have a lot of similarities in our brains compared to other species, and that's illustrated very nicely in this evolutionary figure here. So here we see brains of different mammals, and we can see just by looking at it that there are some of the basic areas that are shared across all of them. For example, primary visual areas, primary sensory-motor areas, and this indicates to us that there seems to be some sort of fundamental principle of anatomical organization that is shared across all these species. And this leads us to the principle of homology. So homology allows us to draw general conclusions about anatomy across species and then to be able to translate those findings across species as well. For example, if we were to conduct an experiment in a macaque monkey in an area which has a homologue in the human brain, then perhaps those results from the macaque study will tell us would give us some implications about what that would mean for the human brain. Important thing to remember is, of course, that all these species are adapted to their own ecological niche, which means that they would have certain cognitive specializations which are specific to them. One example for that is the human brain. Obviously, we have developed one fantastic cognitive feature, which is language. And so the point of this is that we're working out similarities and differences in brain anatomy and function across all the species. We can understand something about what differs and what makes the human brain unique. And in order for us to be able to make such comparative studies, we need to have comparative data set across our two species of interest. So here in the slide, we look on the top row, we see the human brain, and on the bottom row, we see the macaque brain. Now, the macaque brain, aside from the human, is one of the most studied primates in neuroscience, and we have a variety of different measurements that we can compare across the two species. For example, anatomical scans, function, morphology, or that is comparing gyral and sulcal patterns on the cortical surface, connectivity, which I will focus on in this lecture, microstructure, histology, and genetics. Now, this might not be very helpful to you if you're interested, for example, in a species that's not as well studied as the macaque, for example, in the ferret, and you might not have access to all these measurements, and so you might be wondering, well, where do I start, or what measurement is the right one for me? And in this session, we'll focus on connectivity and why that's a very important and good measure. So diffusion connectivity in this context is derived from diffusion MRI, and diffusion MRI has been shown to be relatively reliably consistently acquired in both in vivo and ex vivo scans, which makes it which is why it's so sort of popular in comparative research, and it makes it a useful measurement. So since obviously we're handling animal research, and animal research has certain ethical implication, a lot of researchers in the field aim to adhere to the 3 R's of animal research, so reduce, refine, and replace animal experiments as much as possible. But in light of this, I want to bring your attention to these fantastic initiatives which are allow for researchers across institutes and continents to be able to share all the data they've acquired. For example, we have the PRIMATE database, which contains neuroimaging data of a variety of different primates. We have the Oxford Digital Brain Bank, which has a variety of postmortem scans for different species, the Brain Catalog, and many, many others. And they're available for use, so if you're interested in this kind of research, please look into those. They're fantastic. Now, to circle back onto the meat of this presentation, which is diffusion MRI tractography. Now, here on this evolutionary tree, we see something similar like in the first slide, but we see diffusion MRI data instead of brain maps. And just by glancing at it, we can see that, especially in the placental mammal side of the figure, they seem to be the basic plan seems to be similar across all of them. And to show you an example of this, if we look here at this differentiation between marsupial animals and placental mammals, and we focus on the placental mammals, we can see that all of the brains share this little red stripe here across all of them. So actually the little red stripe is not so little, but it's in fact the corpus callosum, which is a major commissure white matter tract which connects the two hemispheres. But now if you go back to the marsupial side of the evolutionary tree, these are not the best quality scans, but we actually know that the corpus callosum is absent in these animals. And we don't know why that is, but the point of this to illustrate is that there seems to be these major white matter fibers that have remained in the evolutionary tree since the last common ancestor between placental and marsupial animals. And the corpus callosum is just an example. There's multiple other fibers that is also true for. And if you've acquired your diffusion MRI data, how are you able to look into a specific tract? Well, I'll give you a very, very quick abridged crash course of how diffusion imaging works. So essentially, we want to collect multiple different images, which shows the water diffusion signal across a different set of spatial directions. We then apply some mathematical modeling and combine those images into one tractogram, which shows us the primary direction of water flow or white matter fibers in the brain. And if we have something like we see in this figure, then we can use the method of tractography to look into one specific tract. And yeah, in order for us to do that, we obviously need to know a lot of anatomy, and we need to give the algorithm a starting point or a seed, an endpoint or a target, and that reconstructs our streamline. And it's also important for me to mention that those streamlines are not equivalent to actual axons like you would find in tracing data, for example. And it is true that sometimes you can have false positive results. But as long as we are using the same methodology in a comparative context across our species, then we're minimizing the effects of those problems. And so over the last few years in our lab, we've developed recipes for reconstructions of major white matter tracts that are shared across primates. And here show an example of the human brain, macaque, chimpanzee, and gibbon. And all of these tract recipe reconstructions are implemented in the software FSL, which is freely available. And that means that if you have this installed, you can very easily reconstruct these tracts in your own data. And let me show you how you can do that. So for this section of the educational course, I'll show you a little practical of how you're able to visualize these tracts and also how you can construct one tract of your own from scratch. And so what I would like to draw your attention to is in the toolbox FSL, we have this excellent tool called Extract, which essentially is, with this one single line of code, you can reconstruct however many white matter tracts you're interested in from the atlases that we provide. Of course, we have a few prerequisites, so you need to have preprocessed your raw diffusion MRI data, and you'd have a registration space from diffusion to standard species space and standard species space to diffusion. And optionally, you might like a surface reconstruction, which is great for visualization, but I'll also touch on later on how that could be useful in further analysis. And I'm sorry, this was supposed to be a real-life practical, but we've had to make this in a video format. So I'll walk you through how we can visualize the tract. So Extract itself takes quite a long to run. So in this case, I'll just show you what the output looks like. So we have this very handy toolbox called Extract Viewer. So if you have all of this installed, all you need to type in your terminal is Extract Viewer, drag a directory of whichever species you're interested, and you can indicate the species of your interest. And that opens our very handy visualization tool called Fossilize. And here I'm scrolling through a few select tract reconstructions in the human brain. Yeah, you can easily visualize them, and this is just a sample of them. You can, of course, reconstruct only the ones that you're interested in. But let's say that you're not interested in the human brain and you want to draw a tract from scratch. So here I'm showing you the macaque brain. Oh, sorry, I clicked something. Oh no, okay. I'm going to move a little bit forward. So here I've opened macaque brain in standard surface, and for the purposes of the demonstration, we'll be reconstructing the cortical spinal tract. Okay. So first things first, we need to know our anatomy very well. So we know that the cortical spinal tract is the white matter tract that connects primary sensory motor cortex, goes down to the internal capsule, and ends in the spinal cord. So first we need to start with our starting point, also known as the seed. Yeah, and here I changed the color. So we know that our starting point is primary sensory motor cortex, so we need to draw a mask that covers everything. If you remember from your neuroanatomy courses, the kind of more medial side of the mask will be related to sensory motor perceptions of like foot and toe, and as you go more laterally into the cortex, that will be more related to facial movements. But what's important is that you really kind of, you really collect all of the white matter that's in there. And you need to make your folder with your tract, and you need to call your file seed, or that's the starting point. We then, of course, need an endpoint or a target, and you can have multiple ones for more complex tracts, but for our purposes, we're just looking for a target, let's say, in the pons, in the brainstem, and I'm just drawing kind of like a very rough mask of where the pons in the brainstem would be. And similarly to how we saved the seed mask, in the same order we can save it as a target, and that is our endpoint. If we were to run the tractography like this, unfortunately, it will pick up a lot of other things aside from the corticospinal tract, so we need to also implement some exclusion masks. And here we exclude, let's say, everything from parietal lobe onto the occipital lobe, but we also don't want our corticospinal tract to go too much interiorly in the frontal lobe. So our next exclusion mask will be kind of anterior frontal cortex, and since it's not a commissural fiber and we're only looking at the left hemisphere in this case, we want to make sure that we get rid of any crossing fibers to the other hemisphere. We call this mask as an exclude. And based on these conditions, this is what the reconstructed tract looks like. So this is the corticospinal tract starting from primary sensory motor cortex, going down to the internal capsule, and ending in the brainstem. And this is what it looks like on the other side with masks that have been drawn on the other side. I love doing this, but I can imagine if you're not really into drawing masks and neuroanatomy, then I really suggest that you take a look at our extract database, this ORDS has been done for you. But if you're like me and you really love white matter and really love defining white matter tracks, this toolbox presents you with lots of customization, and you can really specify your tract recipes. And also, if you're studying a cool non-human species brain and you'd like to contribute to this atlas, please let me know. We love collecting weird brains. Okay, so we've reconstructed our white matter tracts. So what? What can we do with it? Well, we can look at quite a few different things. Firstly, just a more simple example, if we reconstruct white matter tracts across different species, we can compare them. And in this case, this is a paper by my colleague Nicole Eichert, and she was looking into the arcuate fasciculus, which, as Stephanie mentioned, is a tract which is implicated in language production. And she was curious to see whether this tract is much expanded in the human brain compared to the macaque. And just by looking at the reconstructions, we can see that that's indeed the case. Notably, the human arcuate fasciculus actually goes a lot more into anterior temporal lobe, which is not really the case in the macaque. And here on this side, we see the reconstruction displayed on surface space, where we can see the difference a little bit more clearly. We can take this one step further, and using these white matter reconstruction, create a so-called connectivity blueprint, which essentially allows us to examine the connectivity between each point in the cortical surface to the rest of the brain. And if we have the same sort of comparable tracks across our two species, we can compare two connectivity blueprints, and we can then compare the connectivity blueprints using a measurement called Karl divergence. Karl divergence essentially just compares how similar or different two probability distributions are. And a low Karl divergence value, which in this case, which in our figure here is in green, means that the two probability distributions are more similar to each other. And so in this difference map between macaque and human brain displayed on a human brain, we can see that there's more difference between macaque and human, there's more similarities in macaque and human in primary sensory cortices and in primary visual sensory cortices. In contrast, a high Karl divergence value, which is here in red, means that there's more difference between the two species. And we can see a lot more difference in association cortex, including temporal lobe, inferior parietal lobe, and also inferior frontal cortex. And if you remember from Jungho's slides, and he showed the myelin maps, actually what we see in the myelin map reconstruction is kind of similar to what we see in the Karl divergence here, is that highly myelinated areas seem to be, if we compare the two images, then highly myelinated areas seem to be kind of more similar, whereas areas where there's less myelin seem to be a little bit more different. Now, you can take this even a step further if you're interested in a specific tract and its divisions. So in this case, this is work by my colleague Zilles, and she was interested in this longitudinal tract called the inferior longitudinal fasciculus. Now, in the human brain, the ILF has different divisions, a lateral one and a more medial one. And using the method of Karl divergence, she wanted to see by which of the two tracks, if she included it in the blueprint, would produce a more similar map between the two species. And looking at the results here, we see that the yellow tract, which is the medial division of the ILF, which is shared across the two species, results in a more similar cow divergence maps. But if she introduced the lateral division of the ILF, then the maps showed a lot more difference. And that tells us that this ILF division maybe is a human specific invention or an expansion which has occurred in the human brain that's not present in the macaque. You can also take this a step further, and in this study we were interested in looking at a set of areas, and in this case it was frontoparietal areas, looking into their connectivity blueprints, so which tracts contribute the most to their profile. We wanted to find the best matching areas in the macaque, and in this case we looked at the top 2% most similar areas, which were this four set of frontoparietal areas based on the blueprint of these areas. And we can see that, yes, the connectivity profiles are similar, but there's still a little bit of difference in terms of the dominance of which tract contributes to the profile. So it's quite a diverse technique, and you can use it to look into plenty of different questions. So this was a lot of information, so what are my take-home messages? So comparative neuroscience can allow us to identify overarching principles in neuroanatomy, can help us understand human brain specializations, help us look into the limits of animal model systems, and then diffusion MRI in terms of white matter tract constructions can help us define common features across these species. And then finally, combining diffusion MRI with other modalities allows us to look into neuroanatomical specializations across species. And that's all I have for you. Please let me know if you have any questions.
Thank you, Katrin, for a great work. So we have just plenty much of time for the questions. So if you have any question, please don't feel free to raise your hand, or also maybe you can also use the app to ask your question. Any questions? Or maybe we can just a moment for people to type. Please, go ahead.
Thank you very much for this interesting presentation. You mentioned in the beginning that you are interested in the homology, I think, of the brain. Can you please expand on that and say what do you mean by that? Thank you very much.
Sorry. Yeah, so homology of the brain. So we are interested in, basically, so it's essentially just a principle that allows us to make a direct comparison between the two species. So in order for an area to be considered homologous between two species, they need to have a similar architecture, similar functional and structural outputs, and that can sometimes be a little bit tricky. So for example, macaque primary visual cortex, we could say, is homologous to human primary visual cortex, but then as you go more into kind of the frontal lobe, things get a little bit more blurry and more difficult to define. And so what we mean with this is we are trying to, I guess, as much as possible, find areas that are similar to each other across the species. And in our lab, we focus a lot on the structural similarity, but it's also important to take into context function and myelin and everything like that. But yeah, that's kind of what we meant by that.
So there's other question from software. A recent study from a team in Leipzig recently reported finding in arcuate fasciculus in the chimpanzee brain using tractography. What do you think about that? I think the very recent study from a German group reported arcuate fasciculus in the chimpanzee brain. So do you think maybe how much just maybe arcuate fasciculus is a preserved structure among human, maybe apes or other non-human primates?
Yeah, so the arcuate is an interesting one because in context of tractography, it's actually quite a difficult tract to reconstruct, at least in probabilistic tractography. Now the chimpanzee has I'm not sure I know exactly which paper this refers to, but the chimpanzee does have an arcuate. Obviously, in the human brain, we see, even though chimpanzees are our closest relatives, in the human brain, we see this massive expansion in association cortex, which is generally where the arcuate runs. And so even with our closest relative, we have this big elaboration of the arcuate fasciculus. So I think there are similar tracts, but I think it's in the human there's probably a massive elaboration because of brain expansion and new types of connectivity.
Thank you. So the other question is, have you ever maybe been considered any comparison across different animal with some maybe disease model? Because let's say maybe if we just ever maybe stroke model or maybe glioma model, or the maybe animal disease model, then those maybe I think pathology may impact some diffusion imaging. So maybe I think a person may ask, do you think maybe it's possible to apply this type of comparative just approach if maybe just an animal is coming from maybe disease model?
So the question, if I understand it correctly, is if we've tried a disease-based model compared to tractography. Yeah, I think there's been plenty of work in that in terms of lesions. There's, like I said, macaque is the most studied primate species ever. So there has been a lot of interesting lesion studies there that can be compared to the human. One very fun example that I can think of, well, it's not a fun example, but it's an interesting example, is I mentioned earlier that the marsupial animals don't really have a corpus callosum, and we're not quite sure why that is. And the corpus callosum is a very important tract for interhemispheric communication. Actually, in humans, in a lot of cases where you need to have, for example, epilepsy seizures, when humans have epilepsy seizures, you need to have epilepsy seizure surgery, which actually severs the corpus callosum tract. And yeah, it's definitely an interesting comparison. But yeah, there's lots of work out there that's also based on clinical findings that is quite relevant.
Thank you. So maybe this is the last, maybe here as a question. Are there any available animal data sets other than primate species rather than sheep data set? How much these database covers non-primates?
Yeah, so there's lots of databases out there that I haven't included here. If you're interested, so the Oxford Brain Bank has not only primates, but a variety of other species. Right now, we have quite a lot of carnivore brains, and we're actually looking into that. So that's available. Yeah, that's available to look into. I believe, I don't recall the exact name, but I believe in New Zealand they're doing a lot of studies with sheep brain, and right now they're acquiring lots of fMRI data with them. I think they were working on a database, but I don't know if it's currently available. So there are lots of resources out there that are not primate only. But yeah, Oxford Brain Bank is a good one. Yeah, there's definitely other things out there that is not just primate-centric.
Thank you. Well, the next one is the last one I can pick. So how to just perform comparison between species when there's non-interspecies difference of the white matter tract between species?
Sorry, if I understand the question, how can we compare between species?
Well, yes, how to compare species if there's kind of known interspecies difference about existence of the tract, for example?
Yeah, so what we find is actually that the main body of the white matter tract, at least where it goes, especially in these longitudinal fibers, for example, with the arcuate and the SLF, which is frontoparietal tracts. So the main body of the tract is quite similar across species. Now, what the problem is that obviously the tract ends in different areas, and those areas are not necessarily homologous with each other. So I think we need to be careful when interpreting those results is we can see, for example, which areas the tract connects in generally, but it could be a bit tricky to say that, let's say, arcuate fasciculus in human is homologous to arcuate fasciculus in chimpanzees, because they would connect slightly different areas. So that's why it's also important to look into the, I guess, histology and also function of the areas that you connect. So it's something that we do keep in mind when we make these comparisons.
Great. Thank you again for Katrin.